Mitochondria, structure and function. Now, in this video, we'll learn about the structure and functions of mitochondria. Mitochondria, they are called as the powerhouse of the cell, you already know, and they occupy space in the cytoplasm. They are called as the powerhouse of the cell because they convert energy into forms that are used by the cell. Now, these mitochondria, they come in various shapes. They can be oval, spherical, elongated. And when we see inside the mitochondria, we find there are two membranes. So it's a double membrane structure. Outer membrane, which is porous, and inner membrane, which is folded. And these folds are called as cristae. Now these folds, they increase the surface area for the ATP generation or chemical reactions inside the cell. And here the energy is released is in the form of ATP or adenosine triphosphate. So pyruvic acid and adenosine triphosphate. So ATP is also called as the energy currency of the cell. Now matrix is the ground substance that is filled with enzymes, coenzymes, water and other molecules inside the mitochondria. Mitochondria are the sites of cellular respiration, which finally generates fuel for the cell's activity. So it provides a lot of energy. Now mitochondria, they divide on their own because they have their own genetic material, they have their own DNA, ribosomes, and they are able to make their own proteins. So that is why they are also called as semi-autonomous organelles, because they have their own DNA, ribosomes, and they can make their own proteins. Summary: mitochondria are the powerhouse of the cell. Mitochondria are involved in production of energy, cell division and growth. Plastids, they are present only in the plant cells, and they are rounded, ovoid or disc-shaped structures which contain pigments. Now, on the basis of the color and pigments contained in the plastids, they are classified into three categories: chloroplasts, chromoplasts and leucoplasts. Now, chloroplasts. These are the green colored plastids, and they are found in the aerial parts of a plant. The green color of the plastid is due to a pigment called chlorophyll, and which can trap the solar energy from the sun. Now, food synthesis process called photosynthesis takes place in this structure. And chromoplasts are the colored plastids, and they have yellow, orange or red color, and they are present in the petals of the flowers and ripe fruits. It attracts insects, animals for pollination of the flowers and the dispersal of the fruits and seeds. Leucoplasts are the plastids which do not have any pigment, so they are colorless or white. They are usually found in storage organs and roots. Leucoplasts contain colorless, filamentous and shaped plastids, and they are of different types. They can be amyloplasts, which store starch in the plant cells. They can be aleuroplasts, which store proteins in the cells. And they can be elaioplasts, which collect all droplets of the cortical cells of the orchids. Vacuoles. Vacuoles are fluid-filled membranes, bound spaces that are present in the cell. It is surrounded by a single membrane called tonoplast. In animal cells, they may contain ingested food material and are called food vacuoles. In unicellular animals, they are associated with the maintenance of water balance and acts as excretory organelles also. Vacuoles are also act as storage sacs for solid, liquid. They are present. They are large in the plant cells, and they are small in the animal cells. In some plant cells, 50 to 90% of the cell volume is of vacuoles. Vacuoles are filled with cell sap, which provide turgidity and rigidity of the cell. Amino acid, sugar, organic acid, proteins, they are stored in the vacuoles, and that is only called as the cell sap. And in some unicellular organism, vacuoles expel excess water and waste from the cell. So these are the functions of the cells: to concentrate mineral salts, osmotic pressure, pigments, and Camillo Golgi. Camillo Golgi is the name of the scientist who has discovered Golgi apparatus for the first time. Camillo Golgi was born in Corteno near Brescia in 1843. He used to study medicine, and he was working on nervous system. Actually, while working on nervous system, he was staining individual nerves and the cell structure. And this method of staining was called as the black reaction. Some of you have asked about in the flip learning questions also. And he also got Nobel Prize in 1906 for this discovery of Golgi apparatus. Good morning, students. Today we'll study the structure and functions of Golgi apparatus. Now, Golgi apparatus is an organelle. It is present inside the plant and animal cell, and it is responsible for performing very important function for the cell. Now it's the structure of Golgi apparatus that you are watching on the screen. It is described by Camillo Golgi. It's a membrane-bound vesicle flattened sacs which are arranged parallelly in stalks called cisterns. It is responsible for packaging, dispatching, and material which is synthesized near endoplasmic reticulum. So its main function is transport proteins and lipids, storage, modification, packaging. Everything is done by Golgi apparatus here. So complex sugars made from simple sugars in Golgi apparatus. Now these small channel-like structures, they are called as cisternae. Proteins and lipids are synthesized inside them. It is also involved. Lysosomes are membrane-bound organelles, and membrane-bound sacs, they are filled with digestive enzymes, and these enzymes, which are basically proteins, are made from rough endoplasmic reticulum, that is ribosomes. Now these are the waste disposal systems of the cells, and they help to clean up the cell. Any foreign particle which is entering the cell, all kinds of foreign matters such as bacteria, food, old organelles, they end up in lysosomes. So lysosomes contain strong digestive enzymes which kill all these substances, and they clean up the cell. They also clean the old organelles, like they are killing the mitochondria right now here. And when the cell is damaged, lysosomes they burst the digestive enzymes and digest their own cells, and that is why they are also